Hello, 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 hello. Hmm. Hello, friends. So today we are discussing next topic of our developmental biology, that is reproduction. Now, in last lecture, we discuss different topics such as we discuss differentiation, and in last lecture we discussed the morphogenesis, where we discuss the role of the morphogenes. Now, in that we discuss early development, that is in early development we discuss different points such as we discuss the commitment, then we discuss differentiation, and we discuss morphogenesis. Now, whatever we discuss up to till last lecture, that is the introduction of the developmental biology. Now, in first lecture we discussed that development, it starts from the zygote, and zygote, it is formed after fertilization. So when male pronucleus and female pronucleus, so that is when male gamete, that is sperm, unites with the female gametes, that is egg, there is release of the or transfer of the nucleus from male side into the egg, and then formation of the zygote, and that zygote readily enter into the development, and further developmental process occur. So that is, we discuss that development, it is start from the zygote. So now from today's lecture we will discuss how zygote will form, and then after zygote formation how development occurs. So today we are discussing the type of the reproductions. So different types of reproduction is there, and here we will discuss that there is different advantage, that is some advantage and disadvantage of the different types of reproduction. So reproduction by two ways that we will discuss. One, it's the asexual, and one, that is the sexual way. It is seen in the different organisms such as bacteria, that is prokaryotes, such as plants, and such as animals. So let's start the reproduction topic and the important points in the reproduction topic. So till date, whatever topics are covered, you solved that questions in the 60 number module. So if you have any doubts, you can ask freely. So let's start the topic. Now, reproduction. So reproduction in the organism. So reproduction, it could be by the two ways. One, this is called as asexual reproduction, and second, it's called as sexual reproduction. So these are the two ways by which the organisms they will reproduce. Now what is the sexual and what is asexual reproduction? So here there is, so suppose in this case, first we are discussing the asexual reproduction. So some species reproduce asexually. So we are discussing that which benefits they will get when they reproduce by the asexual reproduction. So we are discussing first asexual reproduction. Now here in the asexual reproduction, single parent involved. So only one parent it is involved in the asexual reproduction, and there is the mitosis which is occur in the asexual reproduction. So there is doubling, it is occur, and due to the doubling here mitosis is occur. So all progenies they form, they are the clones of the parent. So here all progeny are clone of parents. So clone of parents means they are identical. So whatever genome of the parent, that is doubles, and then it is transferred to the progeny. So whatever phenotypic characteristics of parents, that is exactly equal to the progeny. So means progeny is Xerox copy of the parents. All, so which properties of the parent it is transferred, and therefore these are the 100% similar to the parents. And the examples of the asexual reproduction include, so for example, that is, one is the binary fission, and in binary fission here one bacteria which is divide. So we discuss this in the bacteria. So one bacteria divide into the two progeny. So this is parent and these are the progeny. They are clone of the parents. So they are exactly equal. So that is only doubling is there. So at a particular generation time, completion of generation time, the bacteria will double, such as E. coli. So E. coli's generation time is around 20 minutes, and therefore after 20 minutes it doubles. Then binary fission as well as budding, and budding it is occur in the yeast. So here whatever yeast is there, so in yeast there is the budding. So this is the bud formation. So this is also the type of asexual reproduction. Then as well as there is micropropagation, in which we produces the plant, that is in the plant tissue culture, that is also the asexual type of reproduction, as well as tubers, then rhizome, then stem cutting. So these are also the ways of the asexual reproduction. So in asexual reproduction, here there is the mitosis is involved. So here there is the doubling which is occur. So these are the way of the asexual reproduction. And here there is completely identical progenies are formed. So therefore the major way of asexual reproduction, that is the doubling of the organism. Now why species prefer the asexual reproduction? So major role is, what is advantage of the asexual reproduction? Now in the asexual reproduction, here there is doubling of the organism. So for example, here the major advantage is clones are produced. So all progenies are identical, so no variation produced. There is clone production, that is major advantage. All bacteria or all whatever organism is reproduced through asexual methods, they having equal or identical to the parents. Then that is second, rapid proliferation. So they can able to do rapid proliferation because single parent is involved, no second parent is required, there is no mating, and therefore rapid proliferation is there. So that is within small period of time large number of species could be formed from the asexual reproduction. But here this is the advantage of the asexual reproduction. Now disadvantage of asexual reproduction. Now disadvantage, they are more dangerous because the major disadvantage is, so whatever first advantage is, that is his disadvantage, that is clones produce. So this is major disadvantage, and clone produce means all progeny identical, and it is due to, because here there is no variation produced. So variation generation, because here meiosis is not occur, here mitosis is occur. In mitosis there is only doubling, and therefore what happens is that variations are not produced, all progenies are clones. Now clones, they can survive in the normal favorable conditions. So when all factors are normal, or if environmental conditions are favorable, then all progenies will be happily survive because there is no problem. So if environment favorable, then if environment is favorable, then all progeny will survive. So all progeny survive means 100% progeny will be survive because all are equal. But we know that environmental disturbances is the common phenomenon, because there is continuous, there is any one of the environmental factors, it can be affect or it can be becomes the unfavorable, and therefore all environmental factors, they do not remain stable for long period of time, and therefore this is not possible. If environment becomes unstable, if unfavorable, then the all progenies will be die, and therefore here if environment becomes unfavorable, so unfavorable means there is no favorable growth factors they are present, therefore here all progeny die because all are similar. So here therefore, because we know that environmental disturbances is the common phenomenon, there is in some cases there is high rainy season is there, or if there is dry is occur, or if there is pH is change, or is temperature is increase, or there is humidity is increase. So therefore if environment becomes unfavorable, this progeny will be die because they don't have variations, and variations it is not present because they are doing the mitosis, and therefore that is the major disadvantage ki variations is not present. If variation is present, then in environmental disturbances also some species can be grow in that condition or can be sustain that environment, but if all progenies are equal, they cannot sustain these conditions, and therefore they will die. Then here there is the, therefore in this case more than 90% species, so more than 90% species prefer sexual reproduction. So either if species can reproduce asexually, then also it prefer the sexual reproduction because they want the variation. So that is the major disadvantage of the asexual reproduction. Now what is sexual reproduction? So in this case, so mostly in this case the extinction prone species, they are from the asexual reproduction. So most extinct prone species, or the species they have extincted, they are from the mostly this group, that is they reproduce by the asexually and they produce clones, but environmental factors they are unstable and therefore they can be extinct. So that is the major reason or one of the reason from the extinct prone species. Then as compared to asexual reproduction, the role of the sexual reproduction which is preferred by the organism is, so the organism preferred sexual reproduction because the organism want variation. So sexual reproduction. Now here two parents are involved, so two parents involved, then that is male and female both are involved. Then here progeny is not clone of any parent, so no any progeny is clone of any parent because here there is the variation generated. So variation generated, and therefore here it is not possible कि any progeny it is identical or clone of the parent because here variation is generated. So that is major difference between sexual and asexual reproduction. Now here 50% parent give progeny and 50% parent do not give progeny. So males they not give progeny, so not give progeny, but then also their count is maintained. So males they do not give the progeny, but then also their count is maintained because male requires, because sexual reproduction is there, because meiosis is occur, and therefore from male also there is gametes are involved, and therefore male's progeny will be continuously maintained, is either they are not giving the progeny. So only females give progeny, but then also male's count is maintained. So in sexual reproduction, now how variation is generated? So in sexual reproduction, variation is due to, so how variation is generated? So it is due to, because here meiosis occur. So the major reason is meiosis occur, and meiosis it produces the variable gametes. So variable gametes production by the process of the meiosis, and therefore here variation is produced. Now in meiosis the variation, so meiosis occur in the process of gametogenesis, So in the process of gametogenesis, meiosis is occur. Now in gametogenesis, that is the production of the gametes, or this is called as generation of gametes. So generation of gametes, and therefore here there is the variation is produced. Now in gametogenesis, in which process or in which phase the variation is produced? Now in gametogenesis, the variation is produced by the two regions. The major regions are the regions of variation. So first is the first region is, that is independent assortment, and alone independent assortment, that is pairing of the homologous chromosome. So how pairing is occur, and in gamete which pair is transfer, that is play major role, or that is called as independent assortment. So first independent assortment, it produces large variable gametes. Then there is the, we called it as crossing over, or we can be call it as recombination. So that is also the reason for the variation. And after that fertilization, that is after that any one gamete can be fused with the female. So from male one sperm can fuse with the female, and therefore because of the independent assortment and crossing over, the high variation is generated. Now in the independent assortment, how variation is generated? So independent assortment, so independent assortment, the variation is generated because, so here there is the pairing of homologous chromosome, and the transfer pairs into the, and transfer pair into gamete. So on the basis of transferring pairs, which pairs or which homologous chromosomes will pair, and how or in which gametes which pair is transferred, that on the basis of that independent assortment occur. And independent assortment can alone produce large number of variation. So alone generate large number of variable gametes. So independent assortment alone can generate the large number of the variable gametes. Now how it is generated? So here the homologous chromosomes, they pair and the transfer into the gamete. Now here suppose for example in human, so in human around 23 pairs of chromosome, so around 23 pairs of the chromosome, and in the diploid state, so one progeny or one parent here can produce 2 raised to 23 gametes. So from the one individual 2 raised to 23 gametes they are produced in the human. Now here how 2 raised to 23 gametes are produced? Now for example that we are discussing, these different types of gametes, they produced by the only single individual. Now question is that how these types of or different variable gametes are produced. Now suppose and it is due to because we saying that 23 pairs of the chromosomes are present. Suppose we take one example. Suppose if two pairs of chromosome if present, if in human if two pairs of chromosome is present, then what happen? So if two pairs of chromosome, then what happens? So suppose this is one pair, this is second pair, A and A dash, and there is second chromosome. So this is the B and the B dash. So these are the pairs. So A and B, that is the pair. So two pairs of the chromosomes, they are present. Now here, if there is independent assortment in gamete, which pair is transfer? So how much possible combinations are there? So if two pairs of chromosomes is there, then it may be ki in gametes there is transfer of these pairs A with B, or it may be in gametes it can transfer of this pair, that is A and B dash. It may be the transfer of this pair, either A dash B, or it may be there is transfer of this pair A dash and B dash. So how much possible combinations are there? So here combinations are, so this is A, then this is A, this is A dash, this is A dash. Now this is one gamete, and with this gamete there is formation of pair. So that is A can go with the B, so formation of the this gamete, then A can go with the B dash, formation of the this gamete, or A dash can go with the B, formation of this gamete, and A dash can go with the B dash, so formation of this gamete. So if there is the gamete can transfer, so which type of combinations are formed? So A B, that is one combination, A dash A B dash, that is one combination, A dash B, that is one combination, and A dash B dash, that is one combination. So here if two pairs chromosome, so if two pairs of chromosome, then 2 raised to 2, that is 4 gametes form. Means here there is different types of 4 different types of gametes they are formed if there is the two pairs of the chromosome, and therefore here if we see two pairs is there, then 2 raised to 2 different types of gametes they are formed. Suppose if we suppose we take 3 pairs of chromosome present, if 3 pairs of chromosome is present, then now here if 3 pairs of chromosome, then which gametes to be formed? So I will write directly, you just remember this. If 3 pairs, then see here, first gamete it can be formed, that is ABC, correct? So 3 pairs is there, then A dash BC, then one of the combination is A B dash C, and one of the combination is A B and C dash. Okay, so these 4 gametes are possible. Then another gamete A dash B dash C, that is also the possible combination, and A dash B C dash, that is also the possible combination, and A B dash C dash, that is also possible combination, and last combination is A dash B dash and C dash. Means how much different types of gametes they are produced through the independent assortment. So we only the pair the chromosomes, which pair it is transferred into the gametes. So here if 3 pairs of chromosome, then 2 raised to 3, that is 8 gametes form. So is that correct? So correct. That is, if 3 pairs are there, then 2 raised to 3 gametes they are formed. Now see, if 2 pairs, 2 raised to 2 gametes form. If 3 pairs, 2 raised to 3 gametes form. If 4 pairs, 2 raised to 4 different types gametes form. If 5 pairs, 2 raised to 5 different types of gametes form. And if in human 23 pairs, so if in human 23 pairs of chromosome are present, then what happened? So 23 pairs are present, then from male side, so male side there is formation of 2 into, or formation of 2 raised to 23 gametes. And from the female side, formation of 2 raised to 23 gametes, because in human 2, or in sexual reproduction 2 parents are involved. So in human, if 2 parents are involved, male and female, so from both there is formation of the 2 raised to 23, 2 raised to 23 gametes. So total, if 2 parents are involved, because in sexual reproduction 2 parents are involved, so 2 parents involved in sexual reproduction, then here if 2 parents involved, then the gametes they are formed, that is 2 into 2 raised to 23 gametes. They are formed when there is the 2 parents are involved. So this is the variable gametes, they are formed through the sexual reproduction. So how much types of variable gametes they produce through the process of independent assortment, because we are only discussing now independent assortment, till we are not discussing about the crossing over. After crossing over, so uncountable variation is produced. Now this is the independent assortment. Now here there is, after that independent assortment, there is second event, that is called as the crossing over. So second event, it is called, so this is called as the crossing over. And crossing over, which is occur in the first phase, so prophase first of meiosis 1. Here there is the crossing over occur, and due to crossing over, here the crossing over site is variable. So site of crossing over, it is variable, and due to the reason what happen is that any sites there is crossing over is happen, and if in site is variable, then in progeny which genes to be transferred, that is also the variable. And therefore after independent assortment, so independent assortment, uncountable variation is produced, and after crossing over, here there is uncountable variation generated, because here crossing over site is variable, and therefore after the crossing over there is uncountable variation is generated. Now after crossing over, there is third event, and this is called as the fertilization. So there is the fertilization, and in fertilization there is fusion of the male and female gamete. So fusion of male gamete, so any gamete from the male and the female gamete, that is fused. Now here this fusion, that is also the random. because here any gamete of male can fuse with the female. So random fusion of sperm and egg, and therefore any sperm can fuse with the egg, or any sperm can fertilize the egg. And therefore what happens is that here in each time, or in every time, same type of sperm that is not fertilizing egg. So every time different types of variable gamete that is fertilizing the egg, and therefore here the clone production possibilities, they are not possible, or very minor, because, or very negligible, because here variable gametes are produced. So that is the major reason in between sexual and asexual reproduction, ki here in sexual reproduction clones production it is not possible, because large millions of the uncountable variable gametes they are produced, and every time when egg fuse with the sperm, every time new gametes they will fuse, and therefore every progeny is variable. So in progeny there is variation is present, because sexual reproduction is occur. So that is the major reason, and therefore here random fusion is there, and therefore every generation, so in every generation progeny is not same, or progeny is not equal, or the clone, because here variant gametes they are fused. Then, and therefore the cause of variation, it is the major role is the sexual reproduction. Now, so this is the introduction of the sexual and asexual reproduction. Now in sexual reproduction we discuss the process. So one process, that is the producing the variation, it's called as gametogenesis. So one process in the sexual reproduction that play major role in the production of the variation, that is called as gametogenesis. So what is gametogenesis and how it is occur? Now here gametogenesis, there is generation of the gametes. So generation of gametes, and these gametes are generated through the cells, that is called as the germ cells. So here there is the meiosis process that play major role, and generation of gametes, it is through the process of meiosis. So through meiosis gametes are generated. Now which cells they are participate in the gametes formation? So the major cells they are the participate in the gamete formation, these are called as germ cells. They do the gametogenesis. So they do the gametogenesis, and gametogenesis means they enter into the meiosis. So they do meiosis, and the resting cells of our body, these are called as somatic cells, and these somatic cells they do the mitosis. So these are the two types of our cells, that is germ cells and the somatic cells. So somatic cells they form our somatic structure, that is three germ layers, ectoderm, endoderm and mesoderm, they are from the somatic cells, and they will always do the mitosis process. Only germ cells has ability to do the meiosis. So only in gametogenesis the involved cells are, they are the germ cells. Now how gametogenesis is occur? Now in the animals and plants, the germ cells recruitment or separation, they are different. So as compared to plants, in animals germ cells are separated in early development. So in early development germ cells they are separated, and after the gonad formation they transfer into the gonads, and their separation, it is on the basis of some specific proteins. So for example, in animals such as, so for example, in Drosophila, melanogaster, so in Drosophila, germ cells, they are recruited towards posterior side. So here the germ cells are recruited towards posterior side. And in the germ cells recruitment, the major role it is played by some RNA binding proteins. So in this case here, so in Drosophila, so this is the egg of the Drosophila, and that we will discuss in the syncytial specification. So this is the yolk, which is the centrolysical condition, and in Drosophila some cells, so here there is presence of some mRNAs. These are called as Oscar mRNA. So Oscar mRNA, Wasa mRNA, then Nano's mRNA. And what they did, these mRNAs, they are the RNA binding proteins. These are RNA binding proteins, and these RNA binding proteins, these are also called as P granules, and they present in the posterior side. And what they did, some nucleus from the early development, so after we discuss syncytial specification, so here the some nucleus after 5 to 7 or 5 to 6 divisions, some nucleus they translocate towards the posterior side. So this Oscar mRNA recognize 4 nucleus, and these 4 nucleus they transferred into the posterior side and they convert into the pole cells. So these are called as the pole cells, or this is called as the primordial germ cells. So in future these cells they can form the or produce the gametes. So these are called as pole cells. So recruitment of pole cells, it is towards the posterior side, because in Drosophila we discuss this side is the future anterior side and this side is the future posterior side, and therefore the recruitment of germ cells in which side they are present, so that is towards the posterior side. So in Drosophila some mRNA, that is RNA binding proteins, they play major role, and in which the major role it is played by the Oscar mRNA, which is the P granule that play major role. So this is the unfertilized or fertilized egg of the Drosophila. So this is egg of Drosophila after fertilization that we have discussed. So in Drosophila towards posterior side, these pole cells they are recruited, and decision they are taken in the early stages. Then second, in the frog, so second case in frog means in Xenopus. So Xenopus laevis, that species we will discuss in the development in detail. So in frog, germ cells are recruited towards, so germ cells are recruited towards vegetal pole. So they are recruited towards the vegetal pole. Mostly in the endoderm region is there, so towards ventral side these germ cells are recruited. So in this case here, so For example, this is the frog early egg, and suppose there is formation of the tadpole. So this is the tadpole. Now, so this is the early egg. So in the frog, this is early egg, this is the tadpole. So germ cells, they are present in the vegetal pole. So here the germ cells, they are present. Now in the tadpole, they are present in this side. So towards the vegetal pole, ventral side. So these are the germ cells which is present in the frog. So in frog also there is germ cells, they are recruited towards the vegetal pole. Then, and it is separated cells, and they, after gonad formation, they transmit to the gonads, that is testis or the ovary. Then in nematodes, so in nematodes, the germ cells, that is C. elegans, and C. elegans also we will discuss in the detail. So germ cells, they are recruited towards the posterior end. So germ cells are recruited towards posterior end. So towards posterior end. Now so in C. elegans, so here for example, this is the soil nematode, it is present in the soil, and the germ cells, they recruited towards the posterior side. So these are the germ cells, they recruited towards the posterior side, and this is the structure of the C. elegans. So these are the worms, they are present in the soil. So C. elegans, so Cynorhabditis elegans. Then in C. elegans, in the germ cell specification, the major signaling is occur. So here delta notch signaling play major role. So delta notch signaling play major role in C. elegans. Now whatever delta notch signaling is there, so in that there is in delta notch signaling, the cells getting delta signal, they convert into the germ cells. So there are 2 points, that is cells getting delta signal. So in that, cells getting delta signal and the cells not getting delta signal. So cells getting delta signal, they convert into the germ cells. So they form germ cells, and the cells not getting delta signal, they form the somatic cell. So here delta notch signal that decides which cells to be form the germ cells or which cells to be form the somatoplasm or the somatic cells. So on the basis of delta notch signal, that is decided. Then in the C. elegans, so here also in posterior side the germ cells are recruited. So that is, this is the posterior end. The germ cells are recruited, and here in the germ cell specification we will discuss in further lectures P4 stem cell that play major role. So P4 stem cell gives Z2 and Z3 lineage that produces the germ cells. Then in the C. elegans, either germ cells they convert into spermatogonium or the oogonium. So germ cells they convert into spermatogonium or oogonium. So germ cells they convert into spermatogonium or and spermatogonium, or they convert into the oogonium. So, and that is on the basis of the one message, either germ cells convert into spermatogonium or oogonium. So spermatogonium that is produced in the testis and oogonium that is produced in the ovary. Now on the basis of what this is decided? So this is decided on the basis of FEM3 message. So if FEM3 mRNA translated, then there is formation of the spermatogonium. So then spermatogonium form. So spermatogonium form. And if FEM3 mRNA it is not translated, so if FEM3 mRNA not translate, then there is formation of, so then there is formation of the oogonium. So either spermatogonium is formed or the oogonium is formed, that is decides on the basis of the FEM3 message. So FEM3 gene it is regulated at the translational level, and therefore if it is translated then spermatogonium is formed, and if it is not translated then oogonium is formed. And the message gene name it is given on the basis of loss of function experiment, means if in FEM3 gene there is loss of function is there only oogonium is formed, there is no formation of the spermatogonium, because FEM3 contain loss of function or recessive mutation, and therefore if the recessive mutation in FEM3 then feminizing effect is seen, and therefore there the gene given name as the FEM3. So this is the spermatogonium and the oogonium in the C. elegans. Now this is in case of nematode that we discuss. Now in case of higher animals, how the determination of spermatogonium and oogonium it is regulated. So in case of higher animals, so such as humans or mammals, so in case of higher animals, determination of germ cells. So in case of higher animals, how germ cells determination takes place. So in this case, in case of higher animals, there is germ cell determination, it is on the basis of differential expression of the gene. So here differential expression of genes that play major role in the determination of the germ cells. Now that point we have discussed, differential expression of gene. Here we were discussing in the detail. Now differential expression of gene means, suppose if we discuss the first case, that is in human. So suppose in mammals there is XX XY system which is present in the sex determination. So in this case, so if in the XX XY system in mammals, how sex is determined and how germ cell determination is there, that we are discussing. So suppose if XY individual is there, then we call it as the male. So XY is there, and here if Y is there, then there is presence of the Y chromosome. So here Y chromosome is present. If Y chromosome is present, on Y chromosome there is one region is play major role. It's called as SRY region, which is the active region, and from that there is the some genes are expressed. From SRY region there is expression of testis determining factor is expressed. So if Y chromosome is there, then SRY region is present, and SRY region say there is expression of the testis determining factor, that is SOX9. If SOX9 is expressed, then SOX9 will convert bipotential gonad into the testis. So formation of the testis. So testis formation. So bipotential gonad that convert into the human into the testis. Now in either if testis is formed, then through the testis, in testis there is presence of the Leydig cells, and through the testis there is release of the testosterone. So here there is release of testosterone. Through the Leydig cells. Now as testosterone is released, in males the germ cells they convert into, so in testis the germ cells they convert into, or they form the spermatogonium. So there is formation of spermatogonium. So here there is formation of the spermatogonium in the male. Now in this case, if in the male that we have discussed, if in male XY system is present or XY chromosomes are present, then Y chromosome from which SRY region is expressed, SRY SOX9 will express, and there is formation of testis and release of testosterone, convert germ cells into the spermatogonium. Now in case of female, here, so if zygote containing X chromosome, so here this is the female. Now if female is there, there is no presence of Y chromosome. Y chromosome is absent, so SRY region is not present. Now in that case, if there is no SRY region, then in this case here there is the expression of the female specific gene. So here expression of Wnt4a DAX1 genes are expressed. Now as Wnt4a and DAX1 genes are expressed, the bipotential gonad which is convert into the ovary. So formation of the ovary. So there is formation of ovary formation. Now in this ovary there is presence of the theca cells and the internala cells. So ovary is formed. Now in this ovary there is presence of theca cells and the internala cells, and there is follicles from which, so thecal cells, follicles from which there is a release of the estrogen. So here estrogen release. Now as estrogen is released, then the estrogen will convert the germ cells in the female into oogonium. So germ cells, they convert into the oogonium. So formation of the oogonium. So this is the oogonium that is formed. So either in animals, either spermatogonium is formed or the oogonium is formed, and that is decided on the basis of which genes to be expressed. So that is differential expression of gene. So if Y chromosome is present, then SOX9 is expressed, germ cells converted to the spermatogonium, because this is the male, testis is formed. And if the Y chromosome is not present, XX chromosomes are present, then Wind4A express, and if Wind4A express, DAX1 express, then RESPO1 express, then there formation of the female, and female, that is ovary form, and then there is the formation of the oogonium. So that is in the male and female. So in higher animals that we have discussed determination of the germ cells. Now one by one we will discuss the process of the gametogenesis. So first, that is in male there is process of spermatogenesis. So that is generation of male gametes. So synthesis of male gametes. And the male gametes, they are produced in the seminiferous tubules. So they produce in the seminiferous tubules in the testis. So in testis, seminiferous tubules, there is production of the male gametes. Now in this case here, so start point, so that is spermatogonium that is present in the testis, and it continuously maintain its count through mitosis. So maintain balance through mitosis. So it continuously maintain its balance through the mitosis process. Now spermatogonium division start at the puberty stage. So spermatogonium division start at puberty stage. So it division start at the puberty stage. Now in puberty stage, how the division is started of spermatogonium? So that is by the production of the hormone, that is testosterone, but there is activation of spermatogenesis by the anterior pituitary glands, and therefore division is start. So here so spermatogonium division, so at puberty stage, there is release of LH hormone, so high luteinizing hormone, release from anterior pituitary. So luteinizing hormone release from the anterior pituitary. This luteinizing hormone, it activates Leydig cells of the testes. So it acts on Leydig cells of testes, and these Leydig cells, after activation, they activate or release the testosterone. So testosterone releases by Leydig cells. Now this testosterone, this play major role which activate or stimulate spermatogonium division. So it stimulate spermatogonium division. So this is stimulating spermatogonium division. So stimulate or activate. So stimulate spermatogenesis. So this activate the spermatogenesis. So the signal, it is from the anterior pituitary, and then there is the activation of the spermatogenesis. So in spermatogenesis, the spermatogonium enters into the division. So spermatogonium enters into division. So we discussed that the spermatogonium is the unipotent stem cell. So spermatogonium, and this is the diploid, and this is unipotent stem cell. So this is the stem cell. Now it divide by the asymmetric way. So it divide through the asymmetric way, and this spermatogonium, so there is one of the cell is the spermatogonium, so which maintain its own balance, and the second cell, this is called as the primary spermatocytes. So this is primary spermatocyte. This is also the diploid. This is diploid because this is the mitosis process. So here asymmetric cell division produces primary spermatocytes, which is the differentiated lineage cell, and production of the spermatogonium, that is the diploid cell. So maintenance of the tissue balance means it regenerate itself, self renewal is also there, plus it produces the primary spermatocyte. And now primary spermatocytes enter into the spermatogenesis. So this is enter into meiosis. So from this there is initiation of the spermatogenesis. So this is the initial division will start. So initiation of the spermatogenesis. So this is called as sperm meiogenesis. Now what is spermiogenesis? So spermiogenesis, that is synthesis of the sperms. Now the hormones, they regulate spermiogenesis. So hormones regulate spermatogenesis. or required for the spermatogenesis that we are discussing. So there are different hormones that play major role in the spermatogenesis. For example, here the major role it is played by the one, that is luteinizing hormone. And luteinizing hormone, it is released by the anterior pituitary glands, and luteinizing hormone it regulate the spermatogenesis. So it regulate spermatogenesis. So this is the role of the luteinizing hormone. Then second, there is the role of the testosterone hormone. Testosterone, which is produced by the Leydig cells of the testis, and testosterone it activate the spermatogenesis. So it activate spermatogenesis. So major hormone which activate the spermatogenesis or responsible hormone for the spermatogenesis, that is the testosterone. Then there is the third hormone that play major role. It is called as follicle stimulating hormone. And follicle stimulating hormone that play major role. So they activate the Sertoli cells. So follicle stimulating hormone it's activate Sertoli cells, and Sertoli cells of the testis they regulate all the process of the spermatogenesis. So Sertoli cells play major role in the maturation of the sperms. So role of Sertoli cells. So Sertoli cells role is, so Sertoli cells major role, that is maturation of sperm, control by Sertoli cells. Second, provide growth factors and nutrients for maturation of sperm, for sperm maturation. So it play major role in the providing nourishment. So that is called as, in simple term, nourishment it is provided by the Sertoli cells. So in spermatogenesis major central role it is played by the Sertoli cells, as well as it play major role, that is it control sperm or metabolic process in the seminiferous tubule. So that is the major role of the Sertoli cells. Also it convert testosterone into estrogen. So convert testosterone into estrogen, which is required for the sperm maturation. So these are all role of the Sertoli cells. So this is the complete role of the Sertoli cells. So major role it is played by the Sertoli cells in the maturation of the sperm. So that is regulate the process. So provide growth factors, provide new nourishment, provide control metabolic process, convert testosterone into estrogen, that is required for the spermatogenesis. So this is the role of the Sertoli cells. Then, so in sperm maturation, the estrogen, it is converted by Sertoli cells, and then there is the role of the fourth, growth hormone. And growth hormone, that is also regulate metabolic process or metabolic activities in testes. So these are the hormones, they are responsible for the sperm maturation. Then spermatogenesis. So here we discuss there is formation of the primary spermatocyte. So there is formation of primary spermatocyte. Now this primary spermatocyte, it is the diploid condition. Now primary spermatocytes in the seminiferous tubule, it is present in the peripheral side. So cortical region, it is attached to the membrane. So this is the cortical side of the seminiferous tubules. Surrounding peripheral side, it is attached, and they are type 1, and so these are called as type A and type B spermatocytes. So spermatocytes, they are formed. Now primary spermatocyte readily enter into the meiosis 1, and this is the reductional division, in which there is formation of the secondary spermatocytes. So there is generation of secondary spermatocytes. So this is the secondary spermatocyte, and in secondary spermatocytes, these are secondary spermatocytes, these are haploid. So here there is the reductional division is there in meiosis first, and haploid secondary spermatocytes form, and they are connected with the one bridge. This is called as cytoplasmic bridge, which is present in between spermatocytes. Then secondary spermatocytes readily enter into the second meiosis, and it complete the secondary meiosis. Now secondary meiosis, there is formation of the spermatids, and they are also connected with the cytoplasmic bridge. So this is the spermatid, this is spermatids, and spermatids, they are the haploid, and they are also connected with the cytoplasmic bridge, and this cytoplasmic bridge, it play major role in the maturation of the spermatids. So for the maturation, there is requirement of the some proteins, they are coded on the X chromosome. So here in male, in some spermatids, they contain X chromosome, it is taught independently assortmentally, and some spermatid contain Y chromosome. So what happens is that the major genes for the maturation of the sperm, they are present on the X chromosome, and they are expressed on X chromosome, and that proteins they required for all spermatid for maturation, and therefore the all spermatids, they can get the protein which is coded by the X chromosome. That is, whatever protein it is coded by X chromosome, all spermatids, they get this protein and they will mature. For that there is requirement of the cytoplasmic breach. So that is it required for the maturation of spermatids. So all spermatids required the cytoplasmic breach, and if we do some experiment in that we remove or break the cytoplasmic breach, then here it is seen that if the spermatids contain X chromosome, they can mature normally, but the spermatid, they can contain Y chromosome, they cannot mature and they will die because they don't get this protein, and therefore the spermatids required X, the proteins, they are encoded on X chromosome, that is transferred by the cytoplasmic breach. Now after that, these spermatids from cortical peripheral side, when they go into the lumen side, so this is the lumen side where the sperms are mature and formation of the spermatozoa. So here this is peripheral side, this is the lumen side, and in the maturation the major role it is played by the Sertoli cells. So Sertoli cells, they play major role in the maturation. Now here there is the formation of the sperms. So from each spermatid there is formation of spermatozoa. So this is the spermatozoa or the mature sperms. So this is so these are the mature sperm, and here we will discuss the structure. Here there is presence of the acrosome cap where enzymes are present. Then there is presence of the nucleus of the sperm, which is haploid in compacted nature. There is presence of the mitochondria in the large amount in the neck, which is required for the production of the ATP. So these are the spermatozoa. This is called spermatozoa, or they are the sperms. They are produced within the tubules. So from the one spermatogonium there is formation of the 4 gametes, that is 4 sperms, they are the produced. So after that there is disconnection of the cytoplasmic breach, and each spermatozoa is normally mature in the seminiferous tubules. So this is lumen of the spermatozoa, seminiferous tubule, where these sperms they gets mature. Now this is, we discuss the spermatogenesis process. Now after spermatogenesis, whatever sperms they are produced, they contain different regions. So here we are discussing the structure of sperm, that is male gamete. So we are discussing structure of sperm which is produced in the seminiferous tubules and components of the sperms. Now in the structure of sperms, so here the body of sperm, it is divided into the 3 parts. So this is the head part, then so this is head part, Then this is the middle part, that is the neck, and this is the tail part. So that is the flagellum. So this is the structure of sperm. Then in sperm structure there is presence of, first there is the large, around 3/4 of the region having one large enzymatic cap is present, and this is made up from the Golgi. So this is called as the acrosome. So this is the first structure called as acrosome. And acrosome, this is the properties of the acrosome, that is, this is Golgi derived cap. So this is enzymatic vesicle, contain enzymes, and in that vesicle there is presence of hyaluronidase and proteases. They present in the acrosome. So this is the acrosome or acrosome vesicle. So this is the first structure. It is present in the terminal region, and we will discuss this. The acrosome, it is required for the digestion of the jelly coat. Then here there is presence of some G-actin proteins. So G-actin protein, they are present, and G-actin protein, they play major role in the acrosomal process formation. So after fertilization we will discuss, that is, there is extension of the sperm head, so that is due to the G-actin protein. Then there is presence of the haploid nucleus of the sperm. So nucleus of the sperm, which is the haploid, and this is the nucleus, it is in the highly condensed state. So highly condensed state. Condensed means compacted state is present, and it is due to, because having high disulfide linkage in proteins. So there is high disulfide linkage, as well as there is protamines. So the proteins, they are present, they are protamines. So here protamines, they are present, and the protamines form the high disulfide linkage, and they are highly basic proteins. So highly basic proteins, they are called as protamines. They are present, and therefore they tightly associate with the DNA, because if DNA is highly negative charge, proteins are highly positive charge, then positive negative will interact, and therefore the sperm nucleus, it is in the highly condensed or the compacted state that we called. So this is in the high compacted state because there is presence of the protamines. Then here there is presence of the centriole of the sperm, and that centriole, it form the tail region. So this is the centriole, and it form the tail. So tail formation. Then there is presence of the abundant large mitochondria in the neck region. So these are the mitochondria, they present in the neck region. So abundant mitochondria, they present, they are the required for the ATP synthesis. So these are abundant, and required for high ATP synthesis. So energy required for the movement, and therefore so ATP synthesis, it is done by the mitochondria. So that is the role of mitochondria. Then this is the tail, and tail which is made up from the axonema. So this structure, it is called as the axonema. So that is the tail region of the sperm, and axonema, which is the 11 microtubule structure. So having 9 plus 2 microtubule arrangement. So ये 11 microtubule structure axonema is present, and this axonema structure, if we discuss, then in axonema, so suppose we will discussing the axonema structure, how tail is formed. So in axonema structure, in the surrounding there is presence of the doublets. So here there is presence of doublets. So 9 doublets it is present in the surrounding. So this is first, second, third, fourth, fifth, sixth, 7, 8, 9. So these are 9 doublets, they are present in the surrounding, and then in the middle side, so in middle side there is presence of 2 singlets. So in middle side there is presence of the 2 singlets. So this is 1, this is 2. So these are 2 singlets in the middle side. So here these are the 9 doublets. So it is in the peripheral side, and 2 singlets in the surrounding. So these are 9 doublets. So here this is the, for example, 1, 2, 3, 4, 5, 6, 7, 8. So these are the 9 doublets. These are 9 doublets microtubule, and in between center there is presence of the 2 singlets. 2 singlets, and the 9 doublets having 2 arms, that is outer arm and inner arm. So there is presence of dynein. So this is outer arm and the inner arm of the dynein. So this is dynein molecular motor. So dynein molecular motor, which is the negative directed motor, it is present in the tail. So this is dynein molecular motor, and dynein molecular motor having the 2 arms, that is outer arm and the inner arm. So this is the structure of the axonema. So dynein motor, so this is outer arm of dynein motor, this is inner arm of dynein motor. In between this there is presence of the radial spokes. So here there is presence of radial spokes. So this is the, these are radial spoke. So this is the structure of the axonema, having 9 plus 2 arrangement, that is 9 doublets they are present in the outside, and 2 singlets they are present in central. So these are 2 singlets the central, and 9 doublets surrounded by 2 singlets. And this axonema, it is present in normally, example, it is present in the flagella and the cilia. This is present. So flagella and cilia contain axonema structure. So you have discussed this in the cell biology. Now here we have again discussed this. So in axonema there is presence of dynein molecular motor. So this is the structure of the sperm that we have discussed. So in sperm structure we have discussed the acrosome vesicle, which is the Golgi derived sac, contain enzymes, contain hyaluronidase which digest proteoglycans, and the proteases. The actin protein which required for acrosomal process formation, nucleus which is highly condensed and compacted state nucleus, compacted protamine amino acids are present, high disulfide linkage, and highly basic proteins, they are protamine amino acids. Therefore it is in the highly compacted state. Presence of centriole, it form tail, and presence of mitochondria which is abundant, they required for the ATP synthesis. Then presence of the axonema. So tail, it is formed from the axonema, and that axonema is the 9+2 arrangement microtubule structure, and 9 doublets they are surrounded by the 2 singlets. So this is the axonema structure. Now after that, so this is the structure and functions of the sperm, so components. Then next, that is the process of oogenesis. So oogenesis, and in oogenesis there is the synthesis of the egg. So generation of egg or ovum in female ovary. So that is called as the oogenesis. Now oogenesis step that we are discussing. Now in oogenesis, at early stages of development, so at early stages of the development, here the oogonium starts divide. So oogonium division starts, and oogonium, so it enter into the meiosis first. So here suppose this is the oogonium, and this oogonium is diploid, and this oogonium, it giving rise to the one cell which is the oogonium itself. So one cell is the oogonium, and this is diploid because there is the self renewal, but second cell, it gives rise to the primary oocytes. So this is primary oocyte. This is also the diploid stage, primary oocyte. Now as primary oocyte produce, that primary oocyte readily enter into the meiosis first. So it is enter into meiosis first. So as it enter into the meiosis first, here there is first arrest occur. So it enter into the meiosis first, and in meiosis first arrest occur. So first arrest at meiosis first, and this arrest in the G2M checkpoint in diplotin stage prophase first. So this first arrest is occur. Now this first arrest, it is due to there is absence of the luteinizing hormone. So till luteinizing hormone is not present, the primary oocyte remains as it is in the first primary oocyte, that is remains as it is in the meiosis first. So this is the first arrest in the meiosis first, prophase first diplotin stage. Now in this arrest, so this is arrested. Now this arrest, it is removed at the puberty stage. So here, so at puberty stage, so up to puberty stage, primary oocytes arrested in meiosis first. Now after puberty stage, so at puberty stage, so at puberty stage, there is secretion of the luteinizing hormone. So at puberty stage, there is secretion of luteinizing hormone. So there is increased concentration of the, so increase luteinizing hormone concentration. Now as luteinizing hormone is increased, primary oocyte arrest is removed. So there is first arrest is inhibited or removed, and primary oocyte enter or complete its meiosis first. So primary oocyte, so this is primary oocyte, which is the diploid. When it is in the meiosis first, it is arrested. So here meiosis first is completed, and primary oocyte, it's formed the secondary oocyte. But here one cell which is the larger in size, and second cell it is in the smaller in size. So this is the secondary oocyte formation. Now in secondary oocyte, here there is the primary polar body form. So one cell it is the smaller in size, and one cell it is the larger in size, and it is due to because here there is equal karyokinesis but unequal cytokinesis, and therefore one cell it is in the larger in size, and second cell it is in the smaller in size. So this is the formation of primary polar body, and small size will give the its cytoplasmic content to the large size cell, because here one single egg is formed. Then now this secondary oocyte, which is the having haploid cells, this readily enter into the meiosis second. Now this is readily enter into the meiosis second. Now when it enter into the meiosis second, now in meiosis second there is the second arrest. so it is arrested. So in meiosis second there is second arrest. In metaphase 2, in metaphase 2 second arrest is there, and it is due to because high concentration of cytostatic factor. So here there is high concentration of cytostatic factor, and the cytostatic factor is, so that is MOS is the cytostatic factor, and this MOS will do the inhibition of anaphase promoting complex. So it inhibit anaphase promoting. Therefore, the cells remains arrested in the metaphase. They did not complete metaphase, and they will unable to enter into the anaphase because the anaphase promoting complex, it is the inhibited. So this is the role of the MOS. So MOS is the cytostatic factor. So these are the kinases, they inhibit the anaphase promoting complex. Now this is the second arrest which is occur in the metaphase second, and therefore secondary oocyte remains arrested in the metaphase second. Now after that, so these secondary oocytes, they are released by the ovary into the region of the oviduct. So this process, it is called as oogenesis, or this process, sorry, it's called as ovulation. So this process, it is called as ovulation, and in ovulation here there is release of secondary oocyte. So release of secondary oocyte in the infundibulum region, that is region of oviduct near to ovary. So near to ovary in infundibulum there is release of the secondary oocyte. Now because this secondary oocyte can interact with the sperm, and then this secondary oocytes meiosis will get completed. So this is process is called as ovulation. Now after ovulation, now this is in the arrested in the secondary oocyte form, till it is meiosis is not completed. Now here there is the entry of the sperms. So as sperm enter, the sperm will interact with the egg. So here sperm, egg not secondary oocyte interact. So there is sperm and egg interaction, there is activation of the egg. So activation of egg, there is due to sperm and egg interaction, so calcium in the egg release into the cytosol. So calcium in egg cytosol, so transfer into the egg cytosol. So calcium transfer from ER into egg cytosol. So in egg cytosol there is transfer of the calcium, and as calcium is transferred, now calcium it is in the cytosol. So as calcium come outside cytosol, so calcium role in egg cytosol. So as calcium transfer into the egg cytosol, this calcium play major role in the completion of the meiosis. So after sperm interaction, first calcium it is released into the cytosol. Now in cytosol, calcium will play major role, that is in cytosol calcium will activate the some CAM kinases. So calcium activate some calcium dependent kinases. So it activate some CAM kinases. Now as CAM kinases active, this CAM kinases will inhibit. So CAM kinases will inhibit the MOS. So whatever MOS which is inhibiting anaphase promoting complex, this MOS, that is cytostatic factor, which is inhibited by the CAM kinases and it is degraded. Now this is the play major role. So as CAM kinases inhibit MOS, cytostatic factor, MOS is get degraded and activation of the anaphase promoting complex. So anaphase promoting complex gets activates. So anaphase promoting complex get activated. Now as anaphase promoting complex get activated, whatever secondary oocyte it is present in the secondary oocyte form, it complete its meiosis second. So there is completion of the meiosis second and there is formation of secondary polar body. So these are secondary polar body, and now this is secondary oocyte and this is the formation of secondary polar body. Now here also there is the unequal division occur, so formation of the secondary polar bodies, that is 3 small cells they are present, they are called as secondary polar body, and 1 large cell is formed. 1 large cells, this is convert into the egg or ovum in the next. So this is convert into the egg or ovum. So egg or ovum formation, and these secondary polar bodies they will degenerate, and therefore so they enter into the apoptosis, and finally in female there is after 1 oogonium there is formation of single egg or the ovum. Single egg or ovum it is formed, which is the haploid. So this is egg or ovum. So this is formed after sperm interaction. So this is the egg and ovum formation after the fertilization. So this is the oogenesis, so that we have discussed. So that is in oogenesis single egg is formed after the 1 oogonium, but in spermatogenesis 4 sperms they are formed. Now this is the oogenesis. Now difference between spermatogenesis and oogenesis. So spermatogenesis and oogenesis difference. Now in spermatogenesis here and oogenesis here. So in spermatogenesis there is formation of the 4 gametes, that is sperms they are formed after 1 spermatogonium. Then in oogenesis there is formation of the one female gamete, that is egg, after oogenesis. Then spermatogenesis, here 4 sperms, they are motile, and in oogenesis egg जो सिंगल फॉर्मेशन होता है it is non motile. Then sperms, they are small size, but they are equal size, but in oogenesis here the egg is large size, gamete is formed. Then in spermatogenesis here there is no arrest present, so there is no arrest present. In oogenesis here there is 2 arrest present, so 2 arrest present. So one, it is in the meiosis first, so in meiosis first and meiosis second. Meiosis first, prophase first, and meiosis second, metaphase second, arrest is present. Then here in the spermatogenesis that we have discussed, equal nuclear and equal cytoplasmic division. And in oogenesis that we have discussed, equal nuclear but unequal cytoplasmic division, and therefore there is formation of the polar bodies in the oogenesis. So these are the difference between spermatogenesis and oogenesis. So spermatogenesis occur in the testis, oogenesis which is occur in the ovary, that we have discussed. So this is the oogenesis topic that we have discussed. Now in next lecture we will discuss the structure of the egg and then fertilization. So in this lecture we have discussed the topic that is the gametogenesis. In gametogenesis we have discussed spermatogenesis and oogenesis. So thank you.